Ever think about all the tiny microscopic things out there just waiting for a chance to get inside us, like bacteria, viruses, fungi, all that?
Yeah.
It's pretty wild when you think about how much is going on, on a microscopic level.
It really is a constant bombardment, and the fact that we don't get sick more often is a real testament to just how powerful our immune systems are. I mean, they're incredible.
Absolutely. So that's what we're diving into today, the immune system. We've got this awesome material, chapter 43, right?
Mm.
All about how our bodies recognize these threats, these invaders, and then fight back.
Exactly. We'll be looking at the two main types of immunity, innate and adaptive, how they work, how they're different, how they team up, the whole nine yards.
Perfect. So let's jump right in. Concept 43.1, innate immunity. This is like our frontline defense, the stuff we're born with, right?
Right. It's the first line of defense, and it's super fast. It's in all animals, from the simplest to us humans, and it's really good at recognizing and dealing with a broad range of pathogens. It's like a general alarm system.
And it all starts with barriers.
Yeah.
Like I was reading about how insects have these tough exoskeletons like armor
Right.
and chitin, which is in their gut lining and helps protect them. And isn't chitin like the shells of crustaceans too?
It is. And chitin also acts as a barrier against infection. And
Yeah.
they've got lysosome in their digestive system, which we also have, by the way, in things like tears.
Oh, right. Lysozyme. That's like a natural, what is it, antiseptic. Breaks
That's
down bacterial cell walls. And we've got our own set of barriers too, like our skin, obviously, right? And then the mucous membranes in our respiratory system, digestive tract.
urinary tract, reproductive tract, all lined with those mucous membranes. And that mucus isn't just there to be gross. It actually traps those pathogens.
Right. And then those little hairs, the cilia in our airways, are like tiny little brooms sweeping that mucus along out of our system. It's
Mhm.
incredible how much is happening at that level that we don't even realize. And it's not just physical barriers, is it? There's also the chemical defenses.
Right. We've got lysozyme, as we said, in tears and saliva. And the stomach, I mean, that acidity is like a death trap for a lot of those microbes. Even our skin's pH, slightly acidic, is a problem for some pathogens.
Wow. It's like a multi-layered defense system before those invaders even get a chance to say hello.
Exactly. But some, they manage to sneak through. And that's when the cellular defenses come into play. And here we're talking about phagocytosis, which is,
The Pac-Man defense. Those cells that engulf and destroy invaders. So cool. And this happens in both invertebrates and vertebrates, right? Like they mentioned hemocytes in invertebrates.
yes. And in vertebrates, we have neutrophils and macrophages, which are both types of white blood cells.
So what's the difference? They both gobble up bad stuff, right?
They do, but they kind of have different roles. Neutrophils are like the first responders. They're everywhere in the blood, and they're super quick to get to the site of infection. Macrophages, on the other hand,
They're the heavy lifters.
pretty much. They're larger, they live longer, and they can hang out in specific organs, like the spleen, where they're constantly filtering out pathogens and debris from the blood. They're like the cleanup crew.
Gotcha. But how do these cells know what to attack? I mean, it's not like they can see a virus and be like, hey, you're the bad guy.
That's where those toll-like receptors, or TLRs, come in. These receptors are on the surface of these cells, and they recognize molecules that are common to many pathogens but aren't found in our own cells. It's like they can tell friend from foe just by touch.
So like a bacterial flagellum.
Exactly. Or double-stranded RNA, which is found in certain viruses but not in us. These TLRs are basically like an early warning system, and their discovery was huge. It really changed the way we understood the innate immune system. Won the researchers a Nobel Prize, too.
Wow. Well-deserved, right? I mean, figuring out how our bodies do that is pretty incredible. And so it's not just those engulfing cells, right? There are other specialized cells in the innate system too.
Yep. We've got dendritic cells, which are super interesting because they act as a bridge between the innate and the adaptive immune systems. They kind of present pieces of the pathogens to the T cells, which are part of the adaptive system. It's like they're showing the adaptive system what to look for.
Gotcha. So they're the link. And what about eosinophils? I remember reading about those.
Those are great against larger parasites like worms. They release these enzymes that can basically break down those parasites from the outside.
And so they're like the heavy artillery.
Yeah, you could say that. And then we have the natural killer cells, which are pretty amazing.
Natural killer cells. They sound pretty hardcore. What do they do?
So they're different from phagocytes and cytotoxic T cells. They don't engulf or need that antigen presentation thing. They can just recognize infected cells or even cancerous cells that are basically not acting right.
So how do they do that?
They look for the absence of certain markers that healthy cells have or the presence of stress signals. And if they find a cell that's not displaying the right stuff, they're like, okay, you're out of here.
And they just destroy it.
Well, they release chemicals that tell those abnormal cells to self-destruct. It's called apoptosis.
So it's like they send a signal that says, time to die.
Exactly.
Pretty efficient. So those are the cellular defenses. But what about the stuff that's just floating around in our systems? What they call the soluble defenses.
Right, those are important too. We have those antimicrobial peptides, which are like tiny little proteins that can disrupt the membranes of bacteria and fungi.
So they basically make them leak out and die.
Exactly. And then there are interferons, which are unique to vertebrates.
Interferons. I've heard of those. Are those important for fighting off viruses?
They are. When a cell gets infected by a virus, it can release these interferons, which then go to neighboring cells and tell them, hey, there's a virus around here, you better get ready.
So they're like a warning system.
Right. And they tell the neighboring cells to produce antiviral proteins, so if they do get infected, they're better prepared.
Pretty cool, right? And we've actually figured out how to make interferons in the lab, and we use them to treat some viral infections, like hepatitis C.
Wow, so that's a direct application of our understanding of these innate defenses. That's amazing. And then there's the complement system. It's like a whole team working together, right?
Definitely a team effort. It's a bunch of different proteins in the blood, about 30 of them, that work together in a cascade. When they get triggered, usually by something on the surface of a pathogen or by antibodies.
Oh, so there's a link to the adaptive system there, too.
Right, there's some overlap. Anyway, this cascade of proteins can cause the pathogen to burst, or it can help attract phagocytes to the area, or it can even trigger inflammation. It's like a chain reaction that amplifies the immune response. Makes sense. So we've got barriers, those engulfing cells, those killer cells, and all these soluble defenders. What happens when, like, you get a cut or something and those defenses get breached? Isn't that when inflammation kicks in?
Yeah.
You know, all that redness and swelling?
Right. That's the inflammatory response. It's triggered by signals released by damaged tissues or by the cells of the immune system themselves. Macrophages release these things called cytokines, and mast cells release histamine.
Histamine, that's the stuff that makes you itchy, right?
It does. It also makes those blood vessels near the injury dilate, which means they get bigger. And that brings more blood flow to the area, causing redness and warmth. And the histamine makes the blood vessels leaky, so fluid and cells can leak out into the surrounding tissue. And that's what causes the swelling.
So it's all connected. And what about pus? What is pus anyway?
Basically, it's dead white blood cells, mostly neutrophils, those first responders we were talking about, mixed with dead pathogens and tissue debris and fluid. It's the aftermath of that immune battle. It's pretty gross, but it means your immune system is doing its job.
Right, fighting the good fight. And the fluid, it gets drained away by the lymphatic system, right?
Yep. The lymphatic system is like a drainage system for the body. And it carries all that stuff to the lymph nodes, which are like these little filtering stations where the immune cells hang out and try to catch any remaining pathogens.
So they're like checkpoints.
Exactly. And some of those cells, like the dendritic cells we were talking about earlier, they can actually take those pathogens from the lymph nodes and present them to the T cells in other parts of the body, activating the adaptive immune response.
So it's like a chain reaction, starting from that initial injury and leading to a more targeted response. And what about fevers? They happen sometimes when you get a really bad infection, right? Is that related to inflammation too?
It is. When you have a widespread infection, those macrophages and other immune cells can release chemicals that can actually reset your body's thermostat in the hypothalamus. That's the part of the brain that regulates temperature.
So that's why you get a fever.
Right. And there are some theories about why fevers are beneficial. Some scientists think that higher temperatures might make the phagocytes work better or might speed up tissue repair. And it might even slow down the growth of some pathogens.
So it's like turning up the heat to fight off the invaders.
Exactly. But sometimes, and this is serious, sometimes that inflammation can get out of control, and that can lead to something called septic shock.
That sounds scary. What is that?
It's a life-threatening condition that can happen when the inflammatory response goes systemic, meaning it affects the whole body. and it's usually triggered by a severe bacterial infection. What happens is you get a very high fever, your blood pressure drops dangerously low because the blood vessels dilate too much.
So your blood isn't circulating properly.
Right, and that can damage your organs. It's a very serious condition.
And then there's chronic inflammation, which is like a long-term problem, right? Like with diseases like Crohn's disease and ulcerative colitis.
Exactly. Chronic inflammation is when the inflammatory response is stuck in the on position, even if there isn't an active infection. It's like your immune system is constantly attacking your own tissues, and that can cause a lot of damage.
So the innate system is pretty awesome. But pathogens aren't just going to sit there and take it, are they? They've got their own tricks, right?
Oh, absolutely. They've evolved all sorts of clever ways to evade our defenses. It's like an arms race, you know? They adapt, we adapt. It's a constant back and forth.
And the book gave some examples, right? Like some bacteria have those capsules around them that make it hard for the phagocytes to grab them.
Right. It's like they're covered in Teflon. And others, like Mycobacterium tuberculosis, which causes tuberculosis, they can actually survive inside the macrophages. They basically hide out where the immune system can't get to them.
Sneaky little buggers.
Yeah.
Okay, so that was a great overview of innate immunity, our rapid response team. Let's move on to concept 43.2. Adaptive immunity, which is the more specific, targeted system. And this is unique to vertebrates, right?
It is. It's a more recent evolutionary development, and it's really quite remarkable. It relies on this huge diversity of receptors, each one recognizing a very specific part of a pathogen. And what's really cool is that it has memory.
Memory, how so?
So the first time your body encounters a particular pathogen, it takes some time for the adaptive immune system to really get going. It has to find the right cells, activate them, and then make a bunch of copies of those cells. But once it's done that, it remembers that pathogen.
So the next time you encounter that same pathogen.
Boom. The response is much faster and much stronger. That's acquired immunity, and it's what protects us from getting many diseases more than once.
Got it. And the key players in this system are the lymphocytes, the T cells and the B cells. And they both start in the bone marrow, but then the T cells go to the thymus to mature, right?
Exactly. And those natural killer cells we talked about earlier, they're lymphocytes too, but they're part of the innate system.
Right, we covered that. So what triggers the adaptive immune response? Is it those pathogens we were talking about?
Well, specifically, it's antigens. An antigen is any substance that can trigger a response from a B cell or a T cell. It could be a protein from a virus, a piece of a bacterial cell wall, even a toxin, anything that the body recognizes as foreign.
And each lymphocyte has a specific receptor on its surface that can recognize a specific antigen, right?
Exactly. It's like a lock and key. Each receptor fits only one specific antigen.
And it's not the entire antigen that the receptor binds to, is it? It's just a small part of it.
Right. It's called an epitope. It's a specific region of the antigen.
Mm-hmm.
And one antigen can have lots of different epitopes, so it can be recognized by multiple different lymphocytes.
So it's like having multiple targets on the same enemy.
Exactly. Now, B cell antigen receptors and the antibodies they make have a really cool structure. They're Y-shaped proteins with two identical heavy chains and two identical light chains, all held together by these chemical bonds called disulfide bridges.
Disulfide bridges.
Yeah, they're important for the structure. Anyway, each chain has a constant region and a variable region. And it's the variable regions that form the antigen binding site, that part that recognizes the epitope.
So 2 binding sites per receptor.
Right. And when a B cell encounters its specific antigen and gets the right signals, it can turn into a plasma cell.
And those are the antibody factories, right?
Exactly. Plasma cells are basically antibody-producing machines. They churn out these antibodies, which are like soluble versions of the B cell receptor, and they can circulate throughout the body, binding to those specific antigens wherever they find them.
Got it. And the binding between the antibody and the antigen is really precise, right?
Oh, yeah. It's like a perfect fit. It involves these weak bonds that only form when the shapes match exactly.
So what about T cell receptors? They're a little different, right?
They are. They have two different chains, alpha and beta, and they also have variable and constant regions. But T cells don't bind to intact antigens directly like B cells do. They have to have the antigen presented to them.
Presented to them, what does that mean?
It means that the antigen has to be broken down into fragments and then displayed on the surface of a cell by these special proteins called MHC molecules, which stands for Major Histocompatibility Complex. They're like little platforms that hold up those antigen fragments for the T cells to see.
So it's like the cell is saying, hey, T cell, look what I found.
Exactly. And there are two main types of MHC molecules, class I and class II. Class I is found on almost all cells in the body, and it presents fragments of proteins that are made inside the cell. So if a cell is infected with a virus, it'll present viral proteins on its MHC molecules.
Makes sense. So what about class 2?
Class II is mostly found on specialized antigen-presenting cells like dendritic cells, macrophages, and B cells. And they present fragments of antigens that were taken up from outside the cell. So like if a macrophage engulfs a bacterium, it'll chop up those bacterial proteins and present them on its class II MHC molecules.
So the T cells are basically inspecting those presented antigens to see if there's anything suspicious.
Exactly. And there are different types of T cells, too, each with a specific function.
Right, we'll get to that in a minute. But first, I want to talk about this whole idea of receptor diversity. How do we make so many different B cell and T cell receptors? It's not like we have millions of genes for them, right?
We don't. It's actually quite remarkable how we do it. It's a process called gene rearrangement or VDJ recombination.
BDJ.
Yeah. Basically, the genes that code for the receptors are made up of these different segments. There are V segments, D segments, J segments, and C segments. And during lymphocyte development, those segments get shuffled around and combined in different ways.
So it's like a mix and match system.
Exactly. And it's random, so you end up with millions of different possible combinations, each coding for a slightly different receptor.
That's super clever. And it explains how we can have receptors for so many different antigens, even ones we've never encountered before.
Exactly. But with all that random mixing and matching, there's a chance that some of those receptors might recognize our own molecules, our own antigens. That's bad news because it could lead to autoimmunity, the immune system attacking the body itself.
So how do we prevent that?
That's where self-tolerance comes in. As the lymphocytes are developing, they get tested to see if they react to self-antigens, and if they do, they're eliminated.
So it's like a quality control system. Make sure the bad ones don't get out.
Exactly.
Okay, so we've got all these different receptors, and we've gotten rid of the self-reactive ones. What happens when an actual pathogen shows up and its antigens are encountered by the immune system?
That's when clonal selection happens. The antigen binds to the specific lymphocyte that has the matching receptor, and that triggers that lymphocyte to start dividing and making copies of itself.
So it's like finding the right key for the lock and then making a bunch of copies of that key.
Exactly. And some of those copies become effector cells, which are the ones that actually fight the infection. And some of them become memory cells.
Ah, the memory cells. The ones that provide that long-lasting immunity.
Right. Memory cells are long-lived and they hang around in the body waiting for that same antigen to show up again. And if it does, they can mount a much faster and stronger response.
Got it. So the first time you encounter a pathogen, it takes a while for the adaptive immune system to kick in. That's why you might get sick. But the next time...
You're ready for it. The memory cells are there to quickly eliminate the threat.
Awesome. Okay, concept 43.3, adaptive immunity in action. So we know the adaptive system has these two main branches, right? The humoral response and the cell-mediated response.
Right. Humoral is all about antibodies, those soluble proteins we were talking about. They circulate in the blood and lymph, and they're good at dealing with pathogens that are outside of cells.
And the cell-mediated response.
That's where the cytotoxic T cells come in. They can directly kill cells that are infected with viruses or that have become cancerous.
So they're like the assassins of the immune system.
Kind of. They're very specific and very effective at eliminating those problem cells.
Okay, so we got antibodies and cytotoxic T cells. But there's also another type of T cell, right? The helper T cells. They play a pretty important role, don't they?
They do. They're like the conductors of the immune orchestra. They help activate both the humoral and the cell-mediated responses.
So how do they do that?
Well, they have to be activated first. And that happens when they encounter an antigen that's being presented by an antigen-presenting cell, like a dendritic cell or a macrophage. And the antigen has to be presented on a class II MHC molecule. And then there's this other molecule called CD4 that helps stabilize the interaction between the helper T cell and the antigen-presenting cell.
So it's like a three-way handshake.
Pretty much. And once that handshake happens, the helper T cell gets activated and
Right.
starts releasing cytokines, those signaling molecules we were talking about earlier. And those cytokines tell the other cells of the immune system what to do.
Got it. So helper T cells are essential for getting the whole adaptive response going. Now, let's go back to the B cells and antibodies, the humoral response. So a B cell encounters its antigen. Then what?
Well, binding to the antigen is the first step, but it also needs a signal from a helper T cell to really get going. The helper T cell has to recognize the same antigen presented on a class 2 MHC molecule on the B cell itself. It's like a double check to make sure it's a real threat.
So it needs both signals,
Yeah.
antigen and the helper T cell signal.
Exactly. And once it gets both, it goes through this process called clonal selection and differentiation.
Which
It
is,
means it starts dividing and making copies of itself. And some of those copies become plasma cells, which are those antibody factories we were talking about. And some of them become memory B cells, which provide long-term immunity.
got it. And the antibodies, they don't actually kill pathogens directly, do they?
Yeah.
What do they do?
They have several ways of dealing with pathogens. They can neutralize them, which means they bind to the pathogen and prevent it from infecting cells. Or they can opsonize them, which means they coat the pathogen and make it easier for phagocytes to engulf it. And they can also activate the complement system, that cascade of proteins we talked about earlier, which can lead to the destruction of the pathogen.
So they're like the multi-tool of the immune system.
Pretty much. And there are different types of antibodies, too, each with its own specialized function. There's IgA, which is found in mucosal secretions like saliva and breast milk. IgG, which is the most abundant antibody in the blood and can cross the placenta to protect the fetus. IgM, which is the first antibody produced during an infection. IgE, which is involved in allergies and fighting parasites, and IgD, which is mainly found on the surface of B cells.
Wow, that's a lot of different antibodies. Okay, so that's the humoral response. What about the cell-mediated response? That's where the cytotoxic T cells come in, right?
Yeah.
The assassin.
Right. Cytotoxic T cells are really good at dealing with cells that are already infected, especially with viruses, or cells that have become cancerous.
So they're like the internal security force.
Exactly. And they work in a similar way to helper T cells in terms of antigen recognition. They have to recognize their specific antigen, which is presented on a class I MHC molecule on the surface of the infected or cancerous cell. And they also use a different accessory molecule called CD8 to bind to the MHC molecule.
And once they recognize their target, they just destroy it.
Well, they release these toxic proteins that basically poke holes in the membrane of the target cell and then trigger it to self-destruct. It's pretty brutal, but very effective.
Makes sense. So the cytotoxic T cells take care of the infected cells, and the antibodies deal with the pathogens that are outside of cells. And the helper T cells help coordinate the whole thing.
Exactly. And the book had a really good figure summarizing how it all works together. And of course, we can't forget about the memory cells. They're produced for all three types of lymphocytes, helper T cells, cytotoxic T cells, and B cells, so that the next time you encounter that same pathogen, your immune system is ready to go.
Right. The memory cells are key for long-term immunity. And that brings us to vaccination, which is basically a way to trick the immune system into thinking it's been infected so it can build up those memory cells without actually getting sick.
Exactly. It's one of the most important medical breakthroughs in history. And we've come a long way since Edward Jenner first used cowpox to protect people from smallpox. Now we have all sorts of different types of vaccines, inactivated vaccines, live attenuated vaccines, subunit vaccines, even mRNA vaccines.
Right. There are a lot of options, and they've been incredibly successful at preventing diseases, right? Smallpox has been eradicated, and diseases like polio and measles are much less common than they used to be.
Absolutely. But it's important to keep up with those vaccinations because if people stop getting vaccinated, those diseases can come back. We've seen that happen with measles in recent years, which is really concerning.
It's scary how quickly things can change. Okay, so we've talked about active immunity, which is the kind you get from infection or vaccination. What about passive immunity?
Passive immunity is when you get antibodies from someone else, like a baby getting antibodies from its mother through breast milk, or someone getting anti-venom after a snake bite.
So you're getting the antibodies, but your body isn't making them.
Right. It's temporary protection because those antibodies eventually break down. And you don't develop your own memory cells because your immune system wasn't involved in making those antibodies.
Makes sense. And we've gotten really good at using antibodies as tools, haven't we? Like with monoclonal antibodies?
Oh, yeah. Monoclonal antibodies are super useful. They're basically identical antibodies that are made in the lab, and they're incredibly specific. We use them for all sorts of things, from diagnostic tests to cancer treatments. They're really amazing tools.
Super interesting. Okay, so we've talked about all the good things the immune system does. But what happens when it goes wrong? Concept 43.4. Immune system dysfunction.
Right. Sometimes the immune system can overreact or misfire, and that can lead to a whole host of problems like allergies.
Allergies, everyone knows about allergies. Runny nose, itchy eyes.
Exactly. It's basically a hypersensitive reaction to something that's normally harmless, like pollen or dust mites. It involves those IgE antibodies we were talking about earlier. They bind to mast cells, which are these cells that release histamine and other inflammatory
And
chemicals.
that's what causes the allergic symptoms.
Right. And in severe cases, it can lead to anaphylactic shock, which is a life-threatening allergic reaction that can cause your throat to close up and your blood pressure to drop dangerously low.
And what about autoimmune diseases? That's when the immune system attacks your own body.
Right. Exactly. It's like your immune system has turned against you. And there are lots of different autoimmune diseases, each affecting different parts of the body, like lupus, which can affect the skin, joints, and internal organs. Type 1 diabetes, which destroys the insulin-producing cells in the pancreas. Multiple sclerosis, which attacks the nervous system. Rheumatoid arthritis, which causes inflammation in the joints. The list goes on.
And we don't really know what causes autoimmune diseases, do we?
We don't know for sure. It's probably a combination of genetic factors and environmental triggers, but scientists are working hard to figure it out, and there are some promising new treatments that are being developed.
That's good to hear. So what about things like stress and exercise? Do they affect the immune system?
They do. Moderate exercise is actually good for the immune system, but too much exercise or really intense exercise can actually suppress the immune system temporarily.
So it's all about balance.
Right. And stress can also have a negative impact on the immune system. When you're stressed, your body releases hormones like cortisol, which can suppress the immune response. And sleep deprivation can also weaken the immune system.
So getting enough sleep and managing stress are important for immune health, too.
They are. And then there are immunodeficiency diseases, which are conditions where the immune system isn't working properly. And this can be either inborn or acquired.
So inborn would be like something you're born with, right?
Right. It's usually caused by a genetic defect that affects some part of the immune system, like severe combined immunodeficiency or SCID, where people are born without a functional immune system. It's a very serious condition.
And acquired immunodeficiency would be something that happens later in life.
Right. It could be caused by something like HIV, which attacks the helper T cells, those important conductor cells we were talking about, or it could be caused by certain medications that suppress the immune system, like the ones used to prevent organ rejection after a transplant.
So lots of different things can cause immunodeficiency. And finally, let's talk about how pathogens evade the immune system. We mentioned some of those tricks earlier, like those bacterial capsules that make them slippery. But there are other ways too, right?
Oh, yeah. Pathogens are constantly evolving new ways to evade our defenses. It's like a never-ending battle.
And the book talked about antigenic variation, right?
Right. Some pathogens, like the influenza virus, can change their surface antigens, those molecules that the immune system uses to recognize them. So the antibodies that your body made to fight the flu last year might not work against the flu this year because the virus has changed.
So it's like the virus is wearing a disguise.
Exactly. And then there's latency, which is when a virus infects a cell and then goes dormant, hiding out inside the cell without replicating.
So it's like it's sleeping.
Right. And while it's dormant, it's not producing viral proteins, so the immune system can't see it. And then later on, it can reactivate and start replicating again, causing disease. Herpes viruses are a good example of this.
So they're like sleeper agents. And some pathogens can even directly attack the immune system, right? Like HIV.
Right. HIV infects helper T cells, those critical cells that orchestrate the adaptive immune response. And it destroys them, which weakens the immune system and makes people more susceptible to other infections.
That's why people with AIDS get sick so easily. It's
Mhm.
really scary how these pathogens have evolved these ways to avoid our defenses.
It is. But our immune systems are pretty amazing too. They're constantly adapting and evolving to keep up with those pathogens. It's a fascinating and complex
It
system.
really is. And we've just scratched the surface today. But I think this deep dive has given us a much better understanding of how our immune system works, how it protects us, and what can happen when it goes wrong.
Absolutely. It's a system that's worth learning about. It's crucial for our survival.
Couldn't agree more. So for our listeners out there, I encourage you to think about all the things we've discussed today and consider the amazing complexity of this system that's constantly working to keep you healthy. It's truly a marvel of biology. Expert.